Coming to our next chapter, proteins and amino acid chemistry. So, what are proteins? Proteins can be defined as polymers of L-alpha amino acids. So, they are polymers of amino acids. Many amino acids are joined together by peptide bond, and that forms a protein. So basically the type of amino acids in a protein are of L type. So we have two types of amino acids based on the orientation of amino group. In an amino acid we have D amino acids and L amino acids. And in case of proteins in our body, all the amino acids are of L type. So proteins are polymers of L-alpha amino acids. So this is how we can define amino acids, and proteins can be defined as polymers of L-amino acids. Now coming to the elemental composition of amino proteins. So all the proteins, they have carbon, hydrogen, oxygen, nitrogen and some amount of sulfur. So that is the composition of various elements in the protein. So the major constraint is carbon, accounts for 50 to 55%, hydrogen 6 to 7%, and oxygen 19 to 24%. Coming to nitrogen, it accounts for 13 to 19%, and amount of sulfur its content is 4%. It's up to 4%. So this is the elemental composition of proteins. Now coming to amino acids. So we discussed that proteins are the polymers of amino acids. So what is an amino acid? So amino acid is an organic acid having two groups. It has two functional groups. One is amino group and the other one is carboxylic group. So we can define amino acids are the organic acids having two functional groups, amino group and carboxylic group, are defined as the amino acids. So, and they have each carbon in amino acid has four different groups. One it has carboxylic group and the other one is amino group, and there is hydrogen, and the fourth group is represented as R. So all the amino acids, they differ from each other in side chain, that is represented as R. So when you take glycine, in case of glycine the R group is hydrogen, and whereas in case of alanine the R group is methyl group. So all the amino acids, they differ from each other in the side chain, that is represented by R. So as I told you, we have two types of amino acids, D amino acids and L amino acids. So we can compare the configuration with that of our reference sugar, D-glyceraldehyde and L-glyceraldehyde. So you can compare the orientation of groups on D-glyceraldehyde and L-glyceraldehyde, and you compare with that amino acids. If the amino group is on right side, we call it as D amino acid, and if amino group is on left side, we call it as L amino acids. So that is what proteins are, the polymers of L amino acids, where the amino group is on the left side. Yes, now coming to classification of amino acids. Now we have seen what are proteins, what are amino acids. Now we will see how these amino acids are classified. So those amino acids are classified based on their structure, and in that we have subclassifications like amino acids classified with amino acids with aliphatic side chains, amino acids with hydroxyl group, amino acids having sulfur in their side chain, and aromatic amino acids, acidic amino acids and basic amino acids. So when you take amino acids with aliphatic side chain, we have glycine, alanine, valine, leucine, isoleucine. And when you take amino acids with hydroxyl group, where the R group has a hydroxyl group, so that's why we call them as hydroxy group containing amino acids. For example, if you can take serine and threonine, those are the examples for hydroxyl group containing amino acids. And coming to sulfur containing amino acids, in these amino acids have sulfur in their structure, in their side chain, so that's why we call them as sulfur containing amino acids. Like cysteine and methionine, these two are the sulfur containing amino acids. Aromatic amino acids, they have an aromatic ring like phenylalanine, tyrosine and tryptophan. These are called as aromatic amino acids. And acidic amino acids, so when an amino acid has more than one carboxylic group, then we call those amino acids as acidic amino acids. So basically amino acid has one amino group and one carboxyl group. So when it has more than one carboxyl group, then such type of amino acids we call them as acidic amino acids, because there is an additional SD group in those amino acids. So when you see basic amino acids, here also it has one additional amino group. So when there is an additional carboxylic group, we call them as SD amino acids, and when there is an additional amino group, we call them as basic amino acids. So SD amino acids are glutamic acid and aspartic acid, and basic amino acids are lysine and arginine. So this is in brief about the classification of amino acids based on their structure. Now coming to classification based on their polarity. So some amino acids, they have hydrophobic groups in their side chain, and some of them have hydrophilic groups in their side chain. So based on the type of groups present in their side chain, we classified the amino acids into polar and non-polar. Coming to non-polar amino acids, so these are hydrophobic in nature, and those are alanine, leucine, isoleucine, valine. So these are the some important amino acids with non-polar side chain or hydrophobic amino acids. Coming to polar amino acids, these are hydrophilic in nature, and those or examples are serine, threonine, glutamine, aspargine, lysine. So all those are the polar amino acids. So based on the type of group present in the side chain, we classify them into non-polar and polar. And if they are hydrophobic, we call them as non-polar. If they are hydrophilic, we call them as polar amino acids. Coming to another type of classification, till now we have seen classification based on the structure, classification based on their polarity. Now coming to classification of amino acids based on their nutritional value. So out of 20 amino acids, some amino acids are synthesized in our body and some cannot be synthesized in our body. So based on that, we have classified amino acids into different types. Those are two types of amino acids: essential amino acids and non-essential amino acids. If amino acids can be synthesized in our body, so such type of amino acids we call them as non-essential amino acids. And body can synthesize such type of amino acids. And those amino acids which cannot be synthesized in our body and should be supplied in the diet, so such type of amino acids we call them as essential amino acids. So that is the basis for the classification of amino acids into two types: essential and non-essential. So essential are basically they are required but cannot be synthesized in our body, and non-essential are amino acids required and can be synthesized in our body. So that is the basic difference between essential and non-essential. So there are 10 essential amino acids like methionine, phenylalanine, arginine, valine, histidine, lysine, leucine, isoleucine, threonine. So these are called as tryptophan. These are called as essential amino acids. So these are 8 amino acids, and out of the 10 essential amino acids, 2 are classified as semi-essential because these are required in growing children. Otherwise, this can be synthesized in adults. So because these are required for the growing children, we call them as semi-essential amino acids. So those are arginine and histidine. Right. So these 10 amino acids, including semi-essential amino acids, so these are called as essential because they cannot be synthesized in our body. Right. And remaining 10 amino acids are called as non-essential amino acids like glycine, alanine, serine, aspartate, aspargine, glutamate, glutamine. So all those are called as non-essential amino acids. So this is classification of amino acids based on their nutritional value. And coming to another classification of amino acids based on their metabolic fate. So some amino acids, their carbon skeleton acts as a precursor for the synthesis of glucose. So such type of amino acids we call them as glycogenic or glucogenic amino acids. And there are another type of amino acids whose carbon skeleton acts as a precursor for the synthesis of keto fats or ketone bodies. Such type of amino acids are called as ketogenic amino acids. So this is how we can classify the amino acids based on their metabolic fate. Means when they are undergoing metabolism in the cell, their carbon skeleton acts as the precursor for the synthesis of glucose or fats. So if it is glucose, we call them as glucogenic amino acids, and if it is fats, we call them as ketogenic amino acids. So there are some amino acids whose carbon skeleton can be used for both synthesis of glucose and fats. So such type of amino acids are classified as glucogenic and ketogenic amino acids. So here glycogenic or glucogenic amino acids are alanine, aspartate, glycine, methionine and other amino acids. And coming to ketogenic amino acids, we have leucine and lysine are called as ketogenic amino acids. And there are 4 glycogenic and ketogenic amino acids. Those are phenylalanine, tyrosine, isoleucine and tryptophan. These 4 are called as glycogenic and ketogenic amino acids. Leucine and lysine are the ketogenic amino acids, and all the remaining amino acids, they are called as glucogenic amino acids. So this is the classification of amino acids based on their metabolic fate. And coming to some important properties of amino acids, like amino acids are ampholytes because they can donate protons and they can accept protons. So because of the presence of amino group and carboxyl group, they can donate and accept protons. So this nature is called as amphoteric nature. That's why amino acids are called as ampholytes. Right? And coming to another property is Victor ion or dipolar ion. So as they contain both amino group and carboxyl group, at a particular pH, the amino acid carries both positive charge and negative charge. So when they carries both positive and negative charge, we call that type of molecule as Zwitter ion or dipolar ion because it has both the charges, positive and negative charges. So that is the meaning of Zwitter ion or dipolar ion. Coming to isoelectric pH, what is isoelectric pH? So when an amino acid carries both positive and negative charges at a particular pH, and that pH is called as isoelectric pH. So pH at which amino acid carries both positive and negative charges is called as isoelectric pH or isoelectric point. Right? So that is the isoelectric pH, Zwitter ion and ampholytes, why you call amino acids as ampholytes. So in this picture you can see Zwitter ion. So that Zwitter ion is seen at a particular pH. That pH we call it as isoelectric pH. And at that isoelectric pH it carries both positive charge and negative charge. Right? So that's why we call it as Zwitter ion or dipolar ion. And coming to other properties, some important reactions due to carboxylic group. So decarboxylation. So as I told you, amino acid has both carboxylic group and amino group, 2 functional groups. Some properties because of carboxylic group, and one of them is decarboxylation. So decarboxylation is a process where we are removing the carboxylic group, removal of carboxylic group in the form of carbon dioxide. That results in the formation of a corresponding amine is called as decarboxylation. And this type of reactions, they are catalyzed by enzymes called as decarboxylases. So decarboxylases, their function is they remove the carboxyl group as carbon dioxide, and this type of reactions, they require pyridoxal phosphate as the coenzyme. Right? So histidine on decarboxylation forms histamine, tryptophan on decarboxylation forms tryptamine. Right? So this is how amino acids on decarboxylation, they form corresponding amines. And property due to amino group. So transamination. So the name itself indicates the mechanism in this reaction. Transamination. Trans means transfer, amination means so amino group. So there is transfer of amino group. So basically in transamination there is transfer of amino group from amino acid to a keto acid. That results in the formation of a synthesis of a new amino acid and new keto acid. So this process is called as transamination. We are simply transferring amino group from one amino acid to a keto acid. So the keto acid lacks the amino group. So now you are transferring amino group from amino acid to a keto acid, so that after transferring the amino group, amino acid becomes a keto acid, and after accepting the amino group, the keto acid becomes a amino acid. So this is what happens in case of transamination. Simply you are transferring the amino group. It is called as transamination, and the enzymes catalyzing such type of reactions are called as transaminases or aminotransferases. And coming to another reaction due to amino group, oxidative deamination. Right. So deamination basically means removal of amino group. So here you are removing the amino group from the amino acid, right? So that results in the formation of a corresponding keto acid and a ammonia is released. So whatever the amino group you are removing from the amino acid, so that is released in the form of ammonia. And after removing that amino group from amino acid, it results in the formation of a keto acid. Okay? So this is deamination. Deamination means removal of amino group from the amino acid. So there are two types of deamination, oxidative deamination and non-oxidative deamination. So generally, this removal of amino group is coupled with oxidation, we call it as oxidative deamination, and without oxidation we call it as non-oxidative deamination. And in our body, glutamic acid undergoes oxidative deamination so that it is converted into alpha ketoglutaric acid and ammonia. This is a most important reaction that occurs in the hepatocytes in the detoxification of ammonia, so that ammonia, whatever which is formed in the cells, that is transported to the liver in the form of glutamine and glutamate. So that glutamate undergoes oxidative deamination, and where that ammonia is released, and that is converted into a non-toxic form, that is urea. So you can see in this example, so glutamate is undergoing deamination catalyzed by the enzyme called as glutamate dehydrogenase. So that converts glutamate into oxaloglutamate, alpha ketoglutaric acid and ammonia. So this is what oxidative deamination, amino group, removal of amino group occurs. And till now you have seen the properties due to carboxylic group and properties due to amino group. And there are some important properties due to the side chain. So one of them is transmethylation. So the name itself here indicates there is transfer of methyl group. So that is what happens in case of transmethylation. Transfer of methyl group from the donor, that is whatever the methyl group donor is there, that donates the methyl group, and that is transferred to an acceptor. So transfer of methyl group is called as transmethylation, and those reactions are catalyzed by the enzymes called as transmethylases. Right? So this is about transmethylation, transfer of methyl group. So here homocysteine is converted to methionine, where there is transfer of methyl group to the homocysteine, so that it is converted to methionine. Right? So those are the important properties of amino acids. Now coming to the proteins, there are 4 levels of organization of proteins. That is primary structure, we have secondary structure, tertiary structure and quaternary structure. So basically we have 4 types of protein structure, and those are primary, secondary, tertiary and quaternary. Right? Coming to primary structure, what it indicates? So the primary structure indicates the sequence of amino acids in a polypeptide chain. So that is what the primary structure denotes. It denotes the sequence of amino acids in a polypeptide chain. Right? So that's the primary structure. You can see here, so all the amino acids in a polypeptide chain, they are linked by peptide bonds or amide bonds. Right? So here we have 2 ends in the polypeptide chain, one is amino end, one is carboxylic end. So the first amino acid in a polypeptide chain has a free amino group, we call it as amino end, and the last amino acid in a polypeptide chain has free carboxyl group, we call it as a C-terminal amino acid, right? C amino acid, amino terminal, carboxy terminal. So first amino acid is amino terminal and the last amino acid is carboxy terminal amino acid. And all the amino acids in between, they are joined together by a polypeptide by a bond called as peptide bond or amide bond. So that is what the bond which stabilizes the primary structure of a protein, that is called as peptide bond or amide bond. So primary structure example is insulin structure. So insulin has 2 polypeptide chain, A chain and B chain, right? So those A chain and B chain have they are linked together by disulfide bonds. So that is the example for the primary structure of protein. Coming to secondary structure of protein. So primary structure of protein is a sequence of amino acids in a polypeptide chain, and secondary structure of a protein results because of configurational relationship between residues at a distance of 3 to 4 amino acids. So there is configurational relationship between amino acids which are placed at a distance of 3 to 4 amino acids. So there is interaction between these amino acids, and that interaction results in the folding of the polypeptide chain. So that forms a secondary structure of protein. Right? So basically it is because of the interaction between the amino acids which are placed at a distance of 3 to 4 amino acid residues apart. Right? So what are the bonds stabilizing these secondary structure? So secondary structure is stabilized by hydrogen bonds, electrostatic bonds, hydrophobic interactions, and van der Waals forces. So these four types of bonds will stabilize the secondary structure of a protein. Right? So there are different types of secondary structure like alpha helix and beta pleated sheets. Coming to alpha helix, so the name itself indicates it is a helical structure. Here, so this alpha helix has, there are amino acids in case of alpha helix. So the bonds between the amino acids placed at a distance of 3 to amino acids results in the helical structure of secondary structure. So that is nothing but we call it as alpha helix, right? So here proline disrupts whenever a proline is inserted into that alpha helical structure, it prevents the formation of alpha helic structure. So proline is an amino acid. Instead of amino group, there is amino group in case of proline. That's why we call it as amino acid. So the proline prevents the alpha helical structure. So when you see alpha helical structure, so in case of per turn of helix, we have 3.6 amino acids. So per turn of helix, 3.6 amino acids, and each amino acid is at a distance of, the distance between each amino acid is 0.15 nanometers, and the distance traveled per turn of helix is called as pitch height, and the pitch height is 0.54 nanometers. So that's about the important points regarding the alpha helical structure. It is a helical structure, and the distance traveled per turn of helix is 0.54 nanometers, 3.6 amino acid residues per turn, and the distance between each amino acid is 1.5 nanometers. So coming to another type of secondary structure, beta pleated sheets or beta sheets. So when you see alpha helix, it is a helical structure, whereas beta sheets they are like extended polypeptide chains. So they are like sheets. It is a fully extended polypeptide chain. So that's why it is called as beta pleated sheets. So there's a pleations. So that pleations results in another type of secondary structure. We call it as beta pleated sheets. So in case of beta pleated sheets, so we have more than one polypeptide chain. So those polypeptide chains may be arranged in parallel direction, because so parallel or anti-parallel. So when you say parallel, two polypeptide chains, when they start with the same end, for example, one polypeptide chain 1 starts with amino group and ends with a carboxyl group, and the second polypeptide chain also starts with amino group and ends with carboxyl group. Such type of structures we call them as parallel type of beta sheets, parallel beta pleated sheet. If the polypeptide chains, they run in opposite direction, one starts with amino group and the other one starts with carboxyl group, such type of polypeptide chains we call them as anti-parallel. So beta pleated sheets are of two types, parallel, anti-parallel, and there are some proteins like carbonic anhydrase. It has both type of sheets, like it has parallel and anti-parallel, right? So that's about the beta pleated sheets of secondary structure. So these beta pleated sheets are seen, the examples for beta pleated sheets are carbonic anhydrase and silk fibroin. And in case of alpha helix, the example you can say keratin, myosin. So those are the proteins which have alpha helix in their structure. So in this picture you can see protein with alpha helix and beta pleated sheets. So the helical structure is alpha helix and the fully extended sheets like structure is beta pleated sheets. Right. Right. Now coming to the another type of structure, tertiary structure. So we have seen that secondary structure is because of configurational relationship between residues placed at 3 to 4 amino acids apart. Here there is steric relationship between amino acids which are far away from each other. So that results in the another type of a compact structure. A three-dimensional compact structure is called as tertiary structure. So this is also because of relationship between amino acids, but the amino acids are placed far apart from each other. There is a distance between these two amino acids, but there is a steric relationship between these two amino acids. So that results in a three-dimensional structure. We call it as tertiary structure. So most of the proteins in our body, they exist in this form, that is tertiary structure. So most of the biologically active proteins exist in tertiary structure, and tertiary structure of a protein depends upon the sequence of amino acids in a polypeptide chain. So primary structure determines the biologically active form of the structure, that is tertiary structure. If all the amino acids are placed in a correct sequence, we will get the corresponding biologically active protein which has a three-dimensional structure. If there is any mutation in the amino acid sequence, then it affects the tertiary structure of the protein. So that is the significance of sequence of amino acids in a given polypeptide chain. So here you can see examples for hemoglobin and myoglobin are the examples for the tertiary structure of protein. And coming to the fourth level of organization of protein structure, that is quaternary structure. Right? So till now we have seen primary, secondary, tertiary, and the last structure is quaternary structure. So quaternary structure, generally proteins which have quaternary configuration, they have more than one polypeptide chain. Right? So here quaternary structure can be defined as the association of catalytically or functionally active subunits is called as quaternary structure. So there should be association between the subunits. So each polypeptide chain in quaternary structure we call it as subunit. So these subunits, they have catalytical or functional association. So that is called as the quaternary structure of a protein. Right? So this quaternary structure of a protein is stabilized by hydrogen bonds, ionic bonds, and hydrophobic interactions. These are the forces, bonds which stabilize the quaternary structure of protein. So here you can see the primary structure, which is a linear one, and whenever there is a configuration relation between amino acids which are placed at 3 to 4 residues apart, that results in the helical structure of protein, that is called as alpha helix, and other type is beta pleated sheet, and that is secondary structure. And whenever that secondary structure further undergoes looping and winding, that results in a three-dimensional structure. We call it as tertiary structure. And tertiary structure, and we have a quaternary structure, that is called as association of catalytically or functionally active subunits. So it has more than one polypeptide chain. All the polypeptide chains are called as subunits. Example is hemoglobin and lactate dehydrogenase. So hemoglobin has 4 polypeptide chains, alpha 1, alpha 2, beta 1, beta 2. So when all the subunits, all the polypeptide chain are functionally active, so hemoglobin can transport gases. Same way, when you take catalytically active protein, for example, if you take lactate dehydrogenase, it is a catalytically active protein. So that lactate dehydrogenase has 4 polypeptide chains, 4 subunits. When it has all the subunits, then only it can catalyze the conversion of pyruvate to lactate, lactate to pyruvate. So that is catalytically active subunit association, and in case of hemoglobin it is functionally active subunit association. So that is called as the quaternary structure of protein. Coming to classification of proteins based on their function. So each protein has a different function. Based on that function, we can classify proteins like catalytic proteins, means those help in the catalyzing the reactions. Enzymes. So enzymes are the catalytic proteins and contractile proteins. So which are involved in the contraction, like actin and myosin are called as contractile proteins. Structural proteins like collagen, elastin and keratin. So the most abundant protein in our body, collagen, is a type of structural protein. And there are some proteins which are involved in the transport of important compounds, molecules. So those are called as transport proteins. So you can take examples like hemoglobin, albumin, transferrin, transthyretin. So all those are the proteins which are involved in the transport of some compounds. And compounds which are called as some proteins, there are some proteins which regulate the metabolisms. So those are called as the regulatory proteins, or simply you can call them as hormones, right? So all the hormones, they come under regulatory proteins like thyroid hormones, insulin hormone, growth hormone, ACTH. So all those are the regulatory proteins. And genetic proteins, the proteins associated with nucleic acids, they are called as genetic proteins or nucleoproteins. For example, histones. So histones are associated with the DNA. So these type of proteins, we call them as genetic proteins or nucleoproteins. So coming to protective proteins, there are some proteins which protect the organisms from the invading microorganisms or pathogens. So such type of proteins, we call them as protective proteins, and the examples are immunoglobulins. So the function of immunoglobulins is they protect the body from invading microorganisms, right? So this is the classification of proteins based on their function, right? Coming to another classification of proteins based on their composition and solubility, right? So based on this, we can classify proteins into three types: simple proteins, conjugated proteins and derived proteins. So this is based on their composition and solubility. Right, coming to simple proteins. What are simple proteins? So they are very simple because they have only amino acids in their structure. So proteins which have only amino acids are called as simple proteins. Right? Coming to conjugated proteins. What are conjugated proteins? So these conjugated proteins have protein part plus non-protein part. So in addition to protein part, they have some non-protein part. So that this type of proteins, we call them as conjugated protein. Right? And the non-protein part of a conjugated protein is called as prosthetic group. Right? Coming to derived protein. So you take the simple proteins and conjugated proteins and you subject them to hydrolysis. So whatever the products you are getting, those proteins, we call them as derived proteins. So you take a native protein, original protein, and subject it to hydrolysis. You will get products. So those are the derived proteins. So classification of proteins based on shape. So based on the shape of protein, you can classify them into globular proteins and fibrous proteins. If the proteins are oval in shape, spherical in shape, we call them as globular proteins, like albumin, globulin. These are globular proteins because they have a oval shape or spherical shape. And coming to fibrous proteins, these are needle-like structures, proteins, so that's why you call them as fibrous proteins. They are elongated and needle-like structures. Like for example, you can take collagen, elastin, keratin. These are the examples for fibrous proteins. So this is classification of proteins based on their shape. So classification of proteins based on the nutritional value, right? So we have discussed in the classification of amino acids that we have two types of amino acids based on their nutritional value: essential amino acid and non-essential amino acids. And coming to classification of proteins based on the nutritional value, we can have three types of proteins: nutritionally rich proteins, incomplete proteins, and poor proteins. Right? So this classification is based on the presence of essential amino acids. So any protein, if it has all the required essential amino acids in required proportions, so we call such type of proteins as complete proteins or rich proteins. Like for example, you can take casein, albumin. These two proteins are called as complete proteins because this casein and albumin, they have all the essential amino acids. Right? Coming to incomplete proteins. So when a protein lacks one essential amino acid, such type of proteins we call them as incomplete proteins. Like proteins from the pulses, they lack the essential amino acid methionine, and proteins from cereals, they lack lysine. So such type of proteins which lack only one essential amino acids, we call them as incomplete proteins. Coming to the third type of proteins, that is poor proteins. So when a protein lacks more than one essential amino acids, such type of proteins are called as poor proteins. Right? So these poor proteins, they cannot promote growth. So example is zinc. There is a protein called as zinc present in corn. So that lacks the two essential amino acids, and those are tryptophan and lysine. So this tryptophan and lysine both are essential amino acids, and this zinc protein, it is deficient in these two amino acids. So whenever a protein deficient in more than one essential amino acid, we call such type of proteins as poor proteins. Right? So nutritional rich protein, they can promote growth, and incomplete proteins, when you take in combinations like mixed diet, you can take they can promote growth. But when you take poor proteins, these proteins, they cannot promote growth. So that's about the nutritional classification of protein based on the presence of essential amino acids in a protein. Right? Right. So this is the classification of proteins. Now coming to biologically important peptides. So we have seen that proteins are biologically active, but there are some peptides which have some important functions. So such type of peptides, we call them as biologically important peptides. Right? So generally these biologically important peptides, they have 3 to 10 amino acids in their structure. So if a peptide has 3 amino acids, we call it as a tripeptide. If it has 4 amino acids, we call it as tetrapeptide. 6 amino acids, hexapeptide. 7 amino acids, heptapeptide. Like that, based on the number of amino acids, we have different types of peptides. So coming to the tripeptide thyrotropin releasing hormone, TRH, it is a tripeptide. It releases the TSH. So that is the function of thyrotropin releasing hormone. Coming to glutathione, glutathione is also a tripeptide made up of glycine, cysteine, glutamate. So it is a gamma glutamyl cystinyl glycine. It's nothing but glutathione having 3 amino acids. Right? So that is about the glutathione. It is a very important enzyme, tripeptide, which is required in the detoxification of hydrogen peroxide. So in erythrocytes, hydrogen peroxide is produced, so that damages the erythrocyte membrane. So in order to prevent the toxic effect of the adverse effect of hydrogen peroxide which is produced in the erythrocytes, we require this glutathione, reduced form of glutathione. Okay. So that is the significance of glutathione. Coming to another biologically important peptide, oxytocin and vasopressin, antidiuretic hormone. So these are nanopeptides, means they have 9 amino acids in their structure. Coming to angiotensin 1, it has 10 amino acids. It is a decapeptide. So it increases the blood pressure. So it is a vasoconstrictive in nature. So its function is vasoconstriction. Coming to angiotensin 2, it is formed from angiotensin 1. It is the angiotensin 2 it has 8 amino acids in its structure. It also increases the blood pressure. Right? So these are the biologically important peptides. Coming to denaturation of protein. So whenever you are disrupting or disorganizing a protein structure, that results in a denatured protein. You take a native protein, and when you heat it or when you treat it with some chemicals, it loses its structure. Right? So that disorganization of a protein structure is called as denaturation of protein. So whenever a protein loses its structure, it is nothing but it is undergoing denaturation. And those agents which are causing this denaturation, which are responsible for this disorganization of the protein structure, we call those agents as denaturing agents. So we basically have 2 types of denaturing agents: physical agents and chemical agents. The physical agents like heat, high pressure, high temperatures, right? So those are the X-rays, UV rays. So these are the some physical agents which denature the protein structure, and there are some different chemicals like urease. So urease can denature the proteins. So that is about denaturating agents which are involved in the disorganization, unfolding the protein structure. So in denaturation, most of the proteins they lose their quaternary structure, tertiary structure and secondary structure. But primary structure is not affected by denaturation. So that is very important point in denaturation of proteins. All the different levels of organization of protein structure are affected except the primary structure of protein. Right. So whenever you are denaturing a protein, its solubility decreases and it precipitate the precipitation capacity, precipitation of protein increases. Solubility decreases with the increase in precipitation. Apart from that, whenever you denature a protein, the denatured proteins can be easily digested in our body. So whenever you are cooking, so that cooking is nothing but you are denaturating a protein. So whenever you are denaturating protein, that cooking exposes the more sites in a protein so that it can be easily digested by enzymes in our body. So that is the significance of denaturation of proteins. Thank you.